Alright, so this is the second lecture of the molecular biology unit, and in this lecture we're going to be going over DNA replication, mutations, and DNA repair. So first we're going to knock out some conventions here that you got to remember. So if you remember, DNA is double-stranded. We talked about that in the previous lecture. And there's two ends. There's the 5' end of a strand and the 3' end of a strand. And remember, both these strands are complementary to each other. So on the flip side, this is going to be 5', this is going to be 3'. And so what's important to remember is when DNA is being replicated, it's read in the 3' to 5' direction. That's also true for transcription, which we'll talk about in the next lecture. So what we'll talk about, both these strands are going to be replicated at the same time, but just for simplicity's sake, we'll focus on this strand as the template strand. So it's going to be read in the 3' to 5' direction. So we have the newly synthesized strand down here, and it's going to be synthesized in the 5' to 3' direction. So what that means is you're going to be adding bases that are complementary to the template strand like this, all the way down like this. At the same time, you're also going to be synthesizing a new strand up here. So you're going to be reading this strand 3' to 5', and then you're going to have a new strand up here. It's going to be synthesized 5' to 3', and that's how you get your two new strands. Now, for DNA replication to occur, remember you have these base pairs, and you got hydrogen bonds between the bases, and it's a very rigid structure. So what you need to do is essentially pull DNA apart, pull the two strands apart, and what that creates is an origin of replication. And so that allows the DNA replication enzymes to get in here and do what they do. And so what happens is you have DNA synthesis that actually occurs in two directions or bidirectionally. Then they both go out from the origin of replication. And then DNA replication occurs both in a continuous, as you can see here, manner, and then also in a discontinuous fashion, as you can see here. And we'll talk about why that happens in a few slides. Another thing to keep in mind about DNA replication is that it's classified as semi-conservative replication. So what does that mean? So here's your original strands, right? You're going to replicate both of these, generate two new strands, the blue strands here, and what's going to result is you're going to have two DNA double helices. So you're going to have one here and one here. And what semi-conservative means is that you have a strand that's essentially retained, or the template strand, that's going to be paired with the newly synthesized strand. Same thing down here. You got a template strand here in green, and then you have a newly synthesized strand here in blue. And that is how you would define semi-conservative. Versus conservative, where you replicate both template strands and then you generate a new double helix that is both newly synthesized strand, and then you retain the old double helix where are both the template strands. And this has actually been shown to not be the case. And then the third theory here is dispersive replication, which is where people thought that you would replicate the DNA and then you would essentially chop it up and recombine it. So you'd have some of the newly synthesized pieces combined with some of the template strand in really all four of these strands here. And over time, this has just been shown to not be the case and not really occur in nature at all. So the way DNA replication is described is as semi-conservative. Alright, so now we'll talk about the DNA replication process, and to help us do that, we have this nice diagram showing the major players involved in DNA replication. So first, as you remember, DNA has to be unwound, and the enzyme that is responsible for that is helicase, and you can see that in here separating out the two strands. So here's a template strand. Here's the other template strand, and you can see on both sides that both template strands are being replicated at the same time. We'll get more into this part of the diagram here in a minute. And so essentially what helicase does is it pulls the strands apart, and it allows the DNA replication enzymes to gain access and do what they do. Now, unwinding the DNA double helix actually causes significant strain ahead of where helicase has separated the strand. So here's helicase here. So ahead here, there's significant strain going on because you're essentially twisting this structure. And to help with that is an enzyme called topoisomerase 1. And you can see that here. And what it does is it actually causes a break in one of the strands that allows the strand to break free and rotate around the unbroken strand. So it's essentially like untying a knot. So it rotates around like this, and then it can rejoin the two broken ends and then relieve some of that strain. Now, the enzyme responsible for synthesizing the new strands is DNA polymerase. It reads the parental strand or the template strand and then adds bases to the growing daughter strand. And you can see that going on here. You got DNA polymerase here, you got DNA polymerase here. And as you'll note, we're keeping with the conventions here. We're going from the 5' end up here, synthesizing to the 3' end. And as you note, same thing here. It's reading from the 3' end down to the 5' end. Same thing going on here. It's reading from the 3' end. This is the template strand, synthesizing 5' to 3'. So up here, DNA polymerase is going this way. Down here, DNA polymerase is going this way. And down here, it's a continuous fashion. And in this portion here, it's in a discontinuous fashion. And we'll talk about why that occurs in a few slides here. So as we said, DNA polymerase is adding complementary bases to the template strand to synthesize the new strand. And the way it does is by catalyzing addition of 5' deoxyribonucleoside triphosphates. So you can see these guys down here to the free 3' OH. So this is the 3'. So again, this is 5' here. We're going synthesizing 5' to 3'. So here's 3'. This would be 5' here. 3' here. So it comes down and cleaves here, forming this 3' to 5' phosphodiester bond. And then it yields a pyrophosphate, or PPI, and then it actually leaves a 3' OH here on the base that has been added, and this allows for addition of the next base. And so the process continues. You form another 3' to 5' phosphodiester bond. And so if you remember, this is essentially why it's synthesized 5' to 3'. Now there's chemotherapy drugs that actually target DNA replication at this exact mechanism. So they have a modified 3' hydroxyl group that prevents addition of the next base. So this chemotherapy drug would come in here like this, and it would bind as if it was the next base. There's no 3' OH group here, so you can't keep adding bases. And so what happens is then DNA replication ends because you can't synthesize any more of the strand, and that's what's called chain termination. And what that results in is that you can't make any more DNA, so you can't do any more cell replication and cell division, and so hence killing the cancer. And as a result of helicase unwinding the two strands of DNA, what results is there's a leading strand and a lagging strand. So the lagging strand in this case is up here, and then the leading strand is here. And remember, the leading strand is synthesized continuously, the lagging strand is synthesized discontinuously, and we'll talk more in detail about that in a few slides here. Now, first I want to point out though is there's these single strand binding proteins, and it's these purple guys here in the diagram. And they bind to each of these strands that are pulled apart, and it prevents them from coming back together. And that's a process called reannealing, when the DNA strands that are pulled apart come back together. And you want to prevent that from happening because you want to enable primase and DNA polymerase and these other enzymes to come in and do their work to carry out the replication process. Now back to this diagram, you have the origin of replication. DNA replication is occurring bidirectionally. And this figure we've been looking at is kind of a snapshot of this segment right here. And you have a leading strand, which is extending from this primer in red, going continuously, and then you have the lagging strand going discontinuously from multiple primers. Now to really get in and explain this, so the lagging strand, it consists of multiple primers, multiple RNA primers. And what happens is that DNA polymerase comes in and synthesizes short, discontinuous strands of DNA from each of these primers, and that's shown here. Now these short discontinuous strands of DNA are called Okazaki fragments, and these are extended even further by DNA polymerase, similar to how the leading strand is extended. Now what happens is eventually you get to a point where the only thing keeping this from being a continuous strand are these RNA primers. So you need to cut these out. So you have exonuclease, specifically 5' to 3' exonuclease in eukaryotes, which comes in and cuts these out. And exonuclease essentially just means it's cutting out the nucleotides or the bases in this case. In prokaryotes, it's going to be DNA polymerase 1, which has exonuclease activity, and that's going to do that in prokaryotes. So then you have these remaining gaps like this. So you need to fill these in with nucleotides, and that's where DNA polymerase comes in. DNA polymerase is going to come in and add nucleotides to then fill in these remaining gaps. So then now you have a nice continuous, you have it filled in with nucleotides, but we're not done just yet. So you want to look at it on a closer level here. So you had these Okazaki fragments, right? You had these primers that would have been in here. This would have been a strand of RNA. DNA polymerase came in and added each of these bases to come in and replace. Because, for example, you would have had a uracil here because it would have been RNA. So you add in a thymine, and then you just add in these other bases because you had to cut out the whole RNA segment. So now you got the right bases in, but you still have these gaps here. And so you need to seal these gaps, and that's carried out by an enzyme called DNA ligase. So you seal these gaps, and then you have a nice continuous strand of DNA that's ready to go. So now back to this diagram just to give you a quick summary of all the players. So you have helicase that comes in like this, unwinds the two strands. You got to keep the two strands apart, so that's where single strand binding proteins come in. They bind to the strands, keep them from reannealing. Remember, helicase by unwinding causes a lot of strain. topoisomerase comes in and it relieves some of that strain. Now you have DNA primase, which comes in and synthesizes these RNA primers, which then serve as the source of synthesizing DNA segments that are created by DNA polymerase. Remember, on the lagging strand up here, you have a bunch of discontinuous strands that are synthesized from multiple primers. Down here on the leading strand, you have one primer, and then you synthesize a continuous strand like this. So up here, you got these Okazaki fragments. You need to cut out the RNA primers eventually with exonuclease activity. Then you have DNA polymerase come in and add new bases, and then DNA ligase comes in and seals the gaps to create a newly synthesized continuous strand of DNA. And that's DNA replication. So an interesting feature of DNA polymerase is that it actually has the ability to proofread the newly synthesized strand. So just like when you write a paper and then proofread it, it's the same kind of thing. So it proofreads for base pairing errors, such as forming an AC or a GT pair. Now, when a base pair is detected, so say it's coming along like this, detects a base pair like right here, what it does is it actually removes it via its 3' to 5' exonuclease activity. So this would take this out, and then it would insert the correct nucleotide in its place. So in eukaryotes, you have what's called telomerase enzymes. Now, before we explain what a telomerase enzyme does, we have to explain what telomeres are. So telomeres are these repeating sequences, and here's an example of it right here. And it's actually the same sequence in vertebrates. It's TTAGGG. So it's this sequence repeated multiple times on the ends of chromosomes. And you can see that here. So you have TTAGGG here, and then it gets repeated and repeated on both ends of the chromosome. And these repeat sequences called telomeres, they exist to protect chromosomes from damage and prevent them from fusing with each other. So if you have a chromosome like this, if you remember, DNA replication, it starts somewhere in the middle. Remember, the chromosomes unwind, and it starts somewhere in the middle. And then it goes bidirectionally. So it goes in both directions. It doesn't start at one end of the chromosome. It doesn't start like that. And so it goes bidirectionally. And so on either end, you have these telomeres in place to help protect the chromosome and then essentially insulate the genetic material, which is what's most important. Now, the problem with that is with each replication, you lose some of these telomere sequences. So you can see this here. We have some of the sequences overhanging. We've lost some nucleotides down in here. And that's where telomerase comes in. So telomerase comes in and prevents the loss of genetic material with every duplication by adding these repeat sequences back on to the 3' end of the daughter strand beyond the coding region. That's important. Because eventually, if you keep using up these telomere sequences, you're going to get into the genetic material, and then you're going to start losing genetic material, which is going to create problems. So you want to preserve these telomere sequences on the ends. And that's where telomerase comes in. So it prevents the loss of genetic material by adding repeat sequences onto the 3' end of the daughter strand. So telomerase contains an RNA sequence that's complementary to that repeat sequence of DNA in the telomere. And so it easily comes in complementary base pairs with parts of the DNA at the 3' end that's overhanging. And so it comes in base pairs, and it serves as a template for adding bases to the ends of the strand. And you can see that here in the diagram. So here, DNA polymerase comes in and adds the nucleotides in to extend the strand and create a new telomere sequence. And then as you can see, it shifts down, and then you can keep adding on even more bases and fully extending these repeat telomere sequences. Then eventually telomerase will drop off, and then DNA polymerase can come in and fill in the missing bases. And you can see that here we have much less of a loss of material. These bases originally were not here, and so because of telomerase serving as a template for these bases, then DNA polymerase can come in and add in the missing bases. And so it is a process that occurs with each replication, and it serves to, like we said, to preserve these telomere sequences at the ends of chromosomes. So specifically, telomerase is expressed in germline cells, and the purpose of that is to maintain the length of chromosomes in both sperm cells and ovum. And that makes sense because these are going to be the source of genetic material for new embryos, and so you don't want there to be any kind of problems. You want the chromosomes and the DNA to have the highest structural integrity. Versus somatic cells actually do not express telomerase, and this causes shortening of chromosomes with every round of replication. So somatic cells in your body, when they undergo DNA replication and then obviously cell division, their chromosome ends are shortening with each round of replication. So mutations, this is a term that gets thrown around a lot. Let's specifically define what it means. So mutations, they occur when there's a change in the DNA sequence that goes undetected by proofreading mechanisms and then eventually DNA repair mechanisms, and it becomes a part of the cell's permanent genome. That's the key part with a mutation, is there's errors that occur with DNA replication all the time, but more often than not, they're actually caught by polymerase or some of these other DNA repair mechanisms we'll talk about, and then it doesn't matter. It's not a big deal. But sometimes they escape all those mechanisms of protection, and then they become part of the cell's permanent genome. What's bad about that is then they can be replicated in subsequent cell divisions. So here's an example down here just showing you a mutation. So you have this guanine, which you had an adenine put in its place, and so that will throw off the sequence. And the reason why that's important is, so you have DNA, then it gets transcribed through transcription into RNA. And remember, the RNA sequence is complementary to the DNA sequence. So the DNA sequence directly affects the RNA sequence, and then the RNA sequence is translated into protein. And the sequence of protein is determined by the sequence of RNA. And the sequence of protein actually determines the protein's function. So if you have a mutation at the DNA level, it's going to affect the RNA sequence, and then the RNA sequence could potentially affect the protein sequence. And if the protein sequence is affected, then that could affect the protein function and even render the protein defective or unable to carry out its function. And so this is where, if you have a mutation in DNA-based sequences of germ cells, these can be the source of inherited diseases. And often inherited diseases are due to defective proteins because of a mutation that was transcribed into the RNA, and then the RNA was translated into the protein sequence, and that was thrown off, and it resulted in either a missing or defective protein. And the problem with that is these source of inherited diseases that are passed on through the offspring, like we were talking about, and then become integrated in all of their cells. So in addition to these proofreading mechanisms, we have what's called DNA repair mechanisms, and they exist to detect and replace changes in DNA base pair sequences that have escaped the proofreading mechanisms. So it's another layer of protection. And again, the reason for that is they want to prevent these mutations from being passed on in subsequent cell divisions. And so we'll talk about some of these DNA mechanisms over the next few slides. All right, so now we're going to talk about some of these DNA repair mechanisms. First, mismatch repair enzymes. So these recognize structural distortions in DNA, like we show here in the diagram. And these structural distortions specifically that this enzyme recognizes are a result of mismatch bases. So you have an incorrect base here, and then it doesn't base pair properly with this base, so this distortion results. So the enzyme comes in after it recognizes this distortion here, makes a cut in the strand here and here with the incorrect base, and then it removes this entire sequence. Then it's able to fill in that sequence with the correct base. DNA ligase then seals the gap between the adjacent segments of this newly synthesized sequence and the original strand. So that now that the correct base is here, it can base pair with this base, and then that gives you a nice uniform sequence and then a nice correct structure. Now this occurs predominantly in the G2 phase of the cell cycle. So if you're not familiar with the cell cycle, we'll talk a lot about it in chapter 5. But just to give you an overview here, so you have G1, you have S, G2, and then M. So it starts in the G1 phase where it gets ready for DNA synthesis, hence the S phase. That's where DNA replication occurs. Then you get into the G2 phase, which gets you ready for mitosis, which is cell division. So this is a continuous cycle that cells go through to replicate their DNA and then eventually divide. And so it's important to know where some of these mechanisms occur, because that it's just good to know, and then also it's highly tested on exams. So this occurs in the G2 phase. This is important also for chemotherapy drugs, as we'll talk about a lot in chapter 5. So now base excision repair, this is a very specific mechanism. So it consists of identifying and removing a single incorrect base. So you can see that here, you have cytosine that's in place of a thymine here. So the mechanism here is that you have base-specific glycosylase enzyme, which is specific to this repair mechanism. So you have base-specific. So each base has its own glycosylase enzyme that detects and removes the incorrect base. So you'd have one that's specific for cytosine here. It would detect this cytosine that shouldn't be here. It would remove it. So then you have it removed here. And then you have apyrimidine or AP endonuclease, which are also specific to this mechanism of repair. They make nicks or cuts on either side in the strand of where that base was removed. Repair enzymes then come in, they fill in with the correct base, as you can see here, and then DNA ligase helps seal the gaps to bring that new base into the sequence. And this occurs throughout the cell cycle. Base excision repair, it's commonly used to repair spontaneous or toxic deamination of bases. So a good example to illustrate this is that cytosine deaminates to uracil at a constant rate. So this is a naturally occurring reaction. Remember, cytosine is a pyrimidine, and then it can form uracil, which is also a pyrimidine that's found in RNA. And this then can result in a CG pair becoming changed to a TA pair, which you can't have. So cytosine deaminates here to uracil, and then uracil is detected as an unmethylated thymine by the uracil DNA So then uracil is detected as an unmethylated thymine by uracil DNA glycosidase, which then removes it. So that gets you here. Then you have AP endonucleases that nick the strand on either side. Very specific, just on that one base that was removed on either side. Repair enzymes fill in the missing base here, and then ligase helps seal the gaps to bring that new base into the sequence. Nucleotide excision repair. So this is a different mechanism that's not as specific as base excision repair. It recognizes and repairs bulky structural distortions of the DNA alpha helix. And you can see that here. So here's a structural distortion here. So the way this mechanism works is that you have specific endonucleases that nick the damaged strand on either side of the damaged nucleotide. So as a result, that sequence is then removed. Then DNA polymerase comes in and adds the missing nucleotides from that sequence that were removed. And then DNA ligase comes in and seals the gaps between the new sequence and the daughter strand. So then you have a nice uniform strand, as you can see here. This occurs throughout the G1 phase of the cell cycle. Now, this is a mechanism that's commonly used to repair DNA damage from ultraviolet light. And that makes sense because ultraviolet light actually causes significant distortions in DNA structure. An example of that is pyrimidine dimers. And you can see that here in the diagram with thymines. Thymine dimers, they get acted on by specifically UV light. That causes a chemical reaction where they end up bonding to each other. As a result, since these aren't supposed to be bound to each other, they cause significant distortions in the DNA double helix. This is a common mechanism for skin cancers that are caused by long-term exposure to UV light, such as basal cell carcinoma and squamous cell carcinoma. So you have these thymine dimers like this. Again, same thing like we talked about earlier. Endonucleases nick on either side. They remove the sequence out like this. Then you have polymerase that comes in and fills in the correct bases. Ligase seals the gap so that you have now a nice uniform sequence. This is just a table in your book that compares the last three mechanisms we just talked about. So mismatch repair, remember it's only detecting mismatch Watson-Crick base pairs. Those are the only type of errors that it can detect. Base excision repair is very specific. It recognizes specific incorrect bases, and they don't have to be these bulky lesions. And the thing is, though, it can only repair damaged bases that are removed by those specific glycosylases. Nucleotide excision repair, this is able to easily recognize and repair bulky DNA lesions like we just talked about. But then as a result of that, it's less specific than base excision repair. All right, so this is an interesting repair mechanism. It's direct repair by an enzyme called 0,6-methylguanine DNA methyltransferase, or MGMT for short. And it repairs guanine bases specifically that have become methylated by alkylating agents. Now, alkylating agents that are agents that add methyl groups, essentially. And they can actually be carcinogens or chemotherapy. Either way, they're damaging DNA, and I'll explain the distinction in a minute here. MGMT, it catalyzes the removal of this methyl group from guanine. So MGMT comes in and essentially steals this methyl group. So it takes the methyl group and it brings it onto itself, and then essentially freeing up the guanine. And then it essentially takes a hit for the team here because it becomes inactivated once it removes the methyl group. So it removes the methyl group and then it becomes inactivated. So this is important because alkylating agents such as carcinogens, so when they add methyl groups, they can do a number of things. First off, that just damages DNA. And in some ways, the reason why it can cause damage to DNA is, remember, methylation can help turn genes off. And so let's say it's an important gene for a DNA repair mechanism, some other repair, base excision repair or something like that. That's one thing that can lead to cancer because you're not able to, preserve the integrity of DNA. Let's say it's a gene that turns on a protein that would induce apoptosis at the appropriate time. If you turn that off, you see that's very common in cancers, that cells aren't undergoing apoptosis when they're supposed to. Then what results is the cell continues to divide and you have uncontrolled growth. So you can see where methylation can really cause some problems, in addition to just straight up just damaging the DNA, which can cause problems in replication and cell division. So back to this, why this is important: if it's inactivated, it can get depleted. So once it receives this methyl group, it can be depleted. Why that's important is because in the case of chemotherapy, So there's alkylating agents that are chemotherapy that are looking to alkylate DNA in cancer cells. So they're actually trying to damage the DNA within cancer cells. The reason why this is important for attacking DNA in cancer cells is because DNA is being replicated at a rapid rate in cancer cells, because the idea is to, keep dividing and keep dividing and keep growing. And so a lot of DNA repair mechanisms get skipped in cancer cell division and cancer DNA replication. That makes it more sensitive to DNA damage. So by these alkylating agents coming in and damaging the DNA within cancer cells, it actually helps with killing off cancer cells. Bringing us back here, why MGMT is important is MGMT can actually work against those cancer cells. So say the chemotherapy is coming in, it's adding a methyl group to damage the cancer DNA. The MGMT in the cancer cell can then take that methyl group right off. Now, what's important is let's say you're going to have a fixed amount of MGMT in the cell. Let's just say an arbitrary number of 10 MGMTs. This is just to make it simple. Eventually, you're going to run out of these 10, right? And so what you have to do is just continue dosing of chemotherapy. So just essentially overrun it with chemotherapy. And then once MGMT is used up, chemotherapy can then carry out its effect. All right, so the DNA repair mechanisms that we've been describing on the previous slides would be called single-strand repair mechanisms because they only act on one of the strands. So now we're going to talk about some double-strand repair mechanisms. So that when you have double-strand breaks, as in breaks of both strands, there's ways to fix that as well. So the first mechanism we'll talk about is non-homologous end joining, and this involves bringing together the ends of two DNA fragments to repair a double strand break. So it's pretty simple here. You have a double strand break. These enzymes, they bring them back into place so that you have two uniform strands. What's important to know though is not all breaks can be clean, as you can have a break like this where you have overhang like this. And what may happen is that the reason why it's called non-homologous end joining is that these two ends that are brought together, they don't need to be exactly perfectly base paired. So they don't have to be homologous to each other. So let's say you bring them in like this, and then when you've brought them in like this, you maybe, to make sure that it ends up eventually fitting so that you have a nice uniform fix, you may have lost some of the DNA sequence on either side or both sides in the process. And so it doesn't really care. What it really cares about is just getting these two fragments back together and then eventually base pair to where they form uniform. So it may result in, cutting off some here and some here to make sure they fit properly and you get two nice uniform sequences back. So another mechanism is by helicase. So just like in DNA replication, it comes in and it unwinds both ends, so on both ends here. And what it does is it creates these short little strands here, and it allows DNA polymerase to come in, as you can see with the green arrows, and fill in the missing bases. So as you can see here, we filled in the missing bases, and then DNA ligase comes in and seals both these gaps, and you have two nice uniform sequences of DNA. All right, so we're going to finish this out with some clinical pearls. And specifically, we're going to talk about inheriting conditions that involve defective DNA repair mechanisms. And as you can imagine, this puts patients at risk for a number of different disorders, and especially developing certain cancers. So Xeroderma Pigmentosum, this is a result of defective nucleotide excision repair. And so this puts patients at risk for skin lesions. I like to think of it as pigment, skin pigment. So they're specifically at risk for basal cell carcinoma, squamous cell carcinoma, and melanoma. All right, so Lynch syndrome, also known as hereditary non-polyposis colorectal cancer, HNPCC. This is actually a pretty high yield syndrome for board exams, and it's a result of defective mismatch repair. Clinical features, you'll see tumors in the intestines and the ovaries in women that develop earlier in life, usually before the age of 40. So this is an example where you could see someone in their 20s developing colorectal cancer. So these patients, they're at risk for colorectal cancer, and then in women, they're also at risk for ovarian cancer. Ataxia telangiectasia, this is a very high-yield disease for the board exam, and it's a result of a mutation in the ATM gene, which codes for a protein by the same name that's responsible for non-homologous joining. So in this disease, this is defective specifically because of this gene, and unfortunately, they develop a series of horrible neurological symptoms, and then telangiectasias, and then immunodeficiencies. So the big three here are neuro, immune deficiency, and then cancer. So the neurological symptoms, they're going to be cerebellar ataxia, difficulty with fine motor movements, and dysarthria. And this isn't fully understood. One of the theories is that it affects neuron development and nervous system development in utero, but that's not 100% confirmed. That's still an area of research. Telangiectasias of the eyes and skin, and again, this is not fully understood either. And then immunodeficiency, which is caused by low numbers of immunoglobulins and lymphocytes, specifically, B and T cells. And as a result of this, since you're immunocompromised, these patients will present with recurrent viral, bacterial, and fungal infections. And what's thought here is that as a part of the maturation of lymphocytes in the bone marrow, is they actually undergo double-strand breaks on purpose to help with maturation of their function so that they could carry out such functions as producing lymphocytes in the case of B cells. But that maturation is dependent on repairing those double-strand breaks by non-homologous end joining. And so when you have that defective, that doesn't happen, and then that results in dying off of lymphocytes. The other thing that can happen is that fragmented DNA. So you have these DNA, and they undergo double-strand breaks. So you have these fragments of DNA, and they can result in either cell death. They can also result in translocations, where maybe let's say there was another break down here. Let's say this translocates with this, and you fuse and you form another new type of chromosome that then drives cancer cell growth. And that's what puts these patients at risk potentially for, if this happens, that's what puts them at risk for lymphomas and then even leukemias too. Fanconi anemia, this is also defective non-homologous joining. Now this is due to a different gene, because you could ask, well, the last one was due to defective non-homologous joining. Why doesn't this one have the same symptoms? It's a different enzyme. There's multiple enzymes that carry out non-homologous joining. In the previous example, it was the ATM gene. In this example, it's a different gene. And it's specifically a gene that more so affects hematological function. So that's why you see, hence it's in the name, Fanconi anemia, you see progressive anemia, pancytopenia, and then numerous congenital abnormalities. And then these patients are at risk for leukemias, which makes sense. It's a hematological malignancy. Bloom syndrome is a rare inherited disease that results from a mutated DNA helicase, which again results in defective DNA repair. Remember, helicase can be involved in double-strand break repair as well. So these patients, they'll present with short stature, facial malar rash, which is in that butterfly distribution over the cheeks and nose, telangiectasias on the skin, and then immunodeficiency, so they can also present with recurrent infections. These patients are at risk for carcinomas, leukemias, and lymphomas. All right, so that wraps up our discussion of DNA replication, DNA repair, and some of the diseases that are relevant to defective DNA repair.